Hello everyone, welcome back. So from today we are going to start a new series that is the keywords related to the medical biology. So we are going to quickly review the cell organelles, and let's start with nucleus. Nucleus contains the genetic material everyone knows, and inside nucleus we have nucleolus, and that takes part in formation of ribosomal subunits, that is RNA. um, Next up, we have ribosome. These things should be on your tips. Like, oh, you already know this information from your school. So I hope you already know this. We're just quickly revising it. The ribosome, it takes part in protein synthesis, and what part, that is a translation, and it's just a basic information. Uh, this it consists of two subunits. This is present in the prokaryotes. 40s plus 60s is present in the eukaryotes. Next up, we have endoplasmic reticulum. The another thing that you should know, that it is continuous with the nuclear membrane. There are two types of endoplasmic reticulum: the rough one or smooth one, based on the presence or absence of ribosomes. And in the rough, we have, since the ribosomes are present, which gives it a rough look, and it is responsible for protein synthesis. So any cell that synthesizes more protein will have abundance of a rough endoplasmic reticulum. Next up, the smooth endoplasmic reticulum. The obviously smooth, that means the ribosomes are absent, and it mainly takes part in detoxification or synthesis of lipid or steroid. So next up, Golgi complex, obviously packaging, processing, and modification of proteins. Next is mitochondria, that is energy production. The ATP is produced and stored here. The structure of mitochondria, double membrane, outer is smooth, inner is, so obviously mitochondria, the outer is smooth and inner has cristae or inner foldings. Next up, we have lysosomes. It contains the hydrolytic enzymes. And obviously, if these enzymes are released into the cell, whatever the reason, it can lead to the autolysis or death of the cell. Next up, we have centrioles. So it consists of two perpendicularly centrosome. It consists of two perpendicular microtubules. And these microtubules, they help in the formation of spindle during the cell division. We will see in later part of this video. Next up, not so important, proteosome. For it is specialized for degradation of proteins. Lysosome for mainly for digestion or other protective function. That was a general. It is proteasome is more specific. So we all know DNA plus histones, or how many histones? 8 histone proteins. They form a nucleosome, and many nucleosome, they form chromatin, and chromatin condenses to form a chromosome. So chromatid is either of the two strands of a replicated chromosome connected by centromere. So see, this is a centromere, and this whole structure is called a chromosome, but a single arm, it is called a chromatid. And based on the location of centromere, chromosomes can be metacentric, submetacentric, acrocentric, telocentric. So this is the picture. So I told you DNA and 8 histone proteins, they form a one structure that is called nucleosome. Many nucleosome, they form chromatin, and chromatin, when condenses, it forms a chromosome. Now, based on the location of centromere, so this center structure, it is called a centromere. If it is present in the center, the two arms are equal, it is called metacentric. If it is not exactly at the center but near the center, it is called submetacentric. If it is near the end, it is called acrocentric, or if it is at the end, it is called telocentric. So now let's talk about cell cycle, or the cell division. Cell cycle consists of interphase or M phase. Interphase has a G1 phase, S phase, and G2 phase. So G1 phase is mainly for growth. S phase is for replication of DNA. So in this, see, when the cell is dividing, the chromosomes number remains the same, that is 46, but the chromatid number is increased. See, I told you that this is a chromosome. There are two chromatids that are connected in the center by a centromere. Now, when the DNA is replicated, DNA is double. So, to accommodate the more DNA, there is reduplication of the chromatids. So, there are double the number of chromatids, but the whole structure, that is the chromosome, it remains the same. The number of chromosomes remain the same. So, I have written here the 46 chromosomes, but the chromatids here it were 46. Now, there are 92. So in G2 phase, we have tubulin protein synthesis. It is the main protein for formation of mitotic spindle. Next, mitosis, equational division, that is the number of chromosomes remains the same before and after the cell division. The first phase is obviously the prophase. It is the disappearing of other non-important structures, the cell membrane, nucleolus, and other organelles. And the centrioles are placed on the opposite pole. So I told you centriole was a micro, consists of two perpendicularly placed microtubules. It doubles and it is placed on the opposite pole so that in future it leads to formation of mitotic spindle. And obviously the chromosomes, they start to condense. Next, metaphase, all the chromosomes align at the equatorial plate. The chromosomes, so this was the mitotic spindle, and the chromosomes then align at the mitotic, they form a equatorial zone. And here we can see the maximum condensation of chromosomes. So when the chromosomes are condensed to the maximum, it is easy to study the karyotype. So karyotype means basically the study of morphology or the number of chromosomes. So since the chromosomes are condensed maximally in metaphase, all the study, whenever you read in a question to study the karyotype, you have to answer metaphase. Because in prophase, they just start to condense. So next phase, anaphase, the separation of chromatids towards the opposite pole, and telophase, the chromosome decondense, cell membrane and organelle reappear, and then cytokinesis occurs. So yeah, this is the picture. We have all this is what interphase, and this part is the M phase. So interphase, it consists of G1 phase, S phase for DNA reduplication, and G2 preparation for mitosis, basically tubulin synthesis. So the mitotic spindle formation takes place and cell division. Now here you can see a G0 phase that I have not told you about. So now let's talk about this. So G0 phase is basically a resting phase or a gap phase, and it is some cells permanently, they stay in the resting phase, and others, if they are stimulated, they can enter the cell cycle and start the division. So based on their ability to enter G0 phase or not, there are three types of cells, that are labile cells, stable cells, and permanent cells. Labile cells, they multiply constantly, so they almost never go into the G0 phase. Stable cells multiply when needed, the liver cells. So they are in G0 phase, but if stimulated due to various factors, they can enter the cell cycle and multiply. So liver, can regenerate. Next, next we have permanent cells. Permanent cells, that it means that they cannot divide. So neurons, cardiac cells, and skeletal muscle cells, they are permanent cells. They are always in the resting phase or the gap phase. They do not divide or they cannot regenerate. Next, this is a picture just to show you the interphase. Prophase, you can see the organelles are disappearing. Metaphase, you can see there's formation of polarly placed centrioles, mitotic spindle, and all the chromosomes, they line at equatorial zone. Anaphase, that is a movement of sister chromatids towards opposite pole. And obviously telophase, the reappearing of other organelles, and the division, that is formation of two new cells. And in all this scenario, the number of chromosomes remains the same, that is 46. So meiosis, reduction, division. So why do we need that? So see, when the fertilization takes place, two human cells, they fuse, and if such thing happens, so if there's a 46 chromosome in one cell, for example in sperm, and 46 chromosome in other cell, they will fuse together in fertilization, and it will become 92. So in every generation, the number of chromosomes will keep on increasing. So practically, since it's not possible, so the sex cells or the sperm or the ova, they have half the number of chromosomes, that is 23 and 23. So for example, if they fuse together during fertilization, the number of chromosomes is restored. So how can the chromosomes number be half? That is because of meiosis. So meiosis, reduction, division, it is for maintaining the number of chromosomes over the generation. It has two parts, the meiosis 1 and meiosis 2. In meiosis 1, the number of chromosomes is reduced to half, so the cell becomes haploid from diploid. Obviously, it has again prophase, but now this has um, more subphases that you need to know about: the leptotene, zygotene, pachytene, diplotene, and diakinesis. And obviously the crossing over takes place in the pachytene phase of prophase 1. You should know this. And this is important for recombination. Recombination has many benefits. But anyways, next up we have metaphase 1. So here there is a double equatorial plane. So this is, so very quickly, let's tell you the difference between a single equatorial plane and a double equatorial plane in mitosis versus meiosis. So in mitosis, obviously the chromosomes had double the chromatids. Okay, let's take a bigger picture. So this was all the 46 chromosomes, they were aligned in one plane. And now when the anaphase was occurring, that was a movement of sister chromatids towards the opposite pole. They were divided from the center, 1, 2, and 3, and like 46. When this happened, the number of chromosome in each cell was still the same. 1, 2 is just the chromatids were half, but the chromosomes were still the same number all around the cell. Now, since in meiosis 1, a double equatorial plane is formed. So if there's a double equatorial plane, that means 1, 2, 3, 4. So only 23 chromosomes will be aligned in one line and 23 chromosomes in another line. And here, when the division would take place, each cell will get only 23 chromosomes. So this is how the chromosome number is halved in meiosis 1. Why? Because during metaphase, a double equatorial plane is formed. So next, phase 1, obviously the moving of chromosomes to the opposite pole, telophase 1, the reappearance of other organelles. Then meiosis 2 is similar to the mitosis. And obviously, since in here it is equational division, the number is 1 and 1 and only. So in meiosis, from one cell, 4 cells are formed, and in mitosis, from 1 to 2 cells are formed. So yes, we can see the subphases of the prophase 1 in meiosis 1. Leptotene, zygotene, pachytene. You can see the crossing over is taking place. Diplotene, after the recombination, when the chromosomes they start to separate, still a chiasma is still formed. And anyways, the diakinesis, the main one is obviously the pachytene phase in which the crossing over takes place. Next is like the overall picture to give you an idea of meiosis. This is interphase meiosis 1 and this is meiosis 2. Prophase 1, metaphase 1, you can see a double mitotic equatorial plane is formed. So anaphase 1, 23 chromosomes will be there in each cell. And prophase 2, metaphase 2, I told you, is similar to the mitosis. So only a single equatorial plane is formed. Anaphase 1 and telophase. So overall, 4 cells are formed in meiosis. So obviously, let's also discuss a Krok question. Students are studying different stages of gametogenesis. They are analyzing a cell having haploid number of chromosomes. So you know in mitosis, the number never halves, the number remains the same. So if a cell is diploid, it will be diploid only. And haploid only occurs in case of meiosis. So obviously they're talking about meiosis. Haploid means the cell has already undergone the first phase of meiosis. That is why there are haploid number of cells. So obviously they're talking about the second division because first division has already happened. That's why the number of chromosomes is half. That's why the cell is haploid. But also they are saying that it is located in an equatorial plane. Equatorial plane, you know, it happens in metaphase. So the answer is metaphase of the second division.